And people could quiet down and sit down and we'll proceed. Must be Lee who's making the most noise. She's not even here. Okay, good. Lee can shout louder than I can. Okay, so Steve Rosa is back here for this particular purpose from the NIH, where he is now at the Vaccine Research Center after having spent an interminable period in our lab here at Stanford. He knows how many years it was. I don't remember. But it's great to have Steve back because he's able to even carry on experiments just like he used to do.
More years.
I don't know if Tom Evans is here. He was doing experiments with Tom Evans.
Tom had to leave early.
He had to leave. But
he had to get back to a wife and family.
Yeah, he's running an infectious disease department or part of a department at UC Davis. Not Steve, but Tom Evans. Steve is going to give the talk, and it's about glutathione in HIV disease, glutathione replenishment in HIV disease.
That's right.
Okay.
And I'm sorry, I was hoping this microphone would amplify my voice, but I'll try to speak up. Okay, good, I'm glad, because sometimes my voice doesn't carry very far. I'm going to be talking today about glutathione and glutathione deficiency, and particularly about a clinical trial that was conducted in the Herzenberg lab here. It actually was completed a few years ago, but we've had quite a few different results from that trial, and even some new recent results through continued analysis. Of course, glutathione, we're very familiar with at this point, is just a tripeptide, and it's been found to be deficient in a number of different human disease states. This is just meant to give you an idea of the wide spectrum of diseases where it has been found to be deficient. I'm not going to go in detail to most of them. I'm focusing today on HIV disease because our trial was conducted in HIV-infected subjects. But you can see in respiratory distress syndrome, in hepatic diseases, in advanced age, cataracts, and in the intensive care unit, there are several disease states where it's been documented to be deficient. N-acetylcysteine is a precursor to cysteine, which is necessary for the synthesis of glutathione. And N-acetylcysteine is one method of replenishing GSH in the human clinically. NAC has been used very safely in humans for many, many years for the purpose of acetaminophen poisoning. So it has a good track record of being safe and effective for that purpose. So this is a good optimal drug to try for the purpose of replenishing GSH in other disease states. So when we did our trial, we chose NAC as the candidate for that purpose. The trial that we conducted was designed mainly to test one major outcome, to see if NAC administered to a group of HIV-infected subjects could replenish GSH. That was not proven before, and it needed to be definitively proven at the beginning level before people could seriously consider this as a treatment. And there were actually many scientists who were even questioning whether NAC was bioavailable. Of course, when you're talking about bioavailability in NAC, it's very questionable because NAC itself is very quickly deacetylated. So even though technically it's not very bioavailable, we like to think of the functional bioavailability of NAC in terms of being able to supply the cysteine necessary for GSH replenishment. Therefore, the outcome measure that we're looking for is the elevation in GSH levels in a group of people who started out to be depleted in GSH. So this slide is to remind me to tell you what the design of the trial was. We chose GSH deficient subjects to start out with because we wanted to start out to make sure we gave ourselves the best chance of seeing an increase in GSH. And I'll briefly mention how we determined whether or not they were deficient in a later slide. The trial itself was fairly short. It was 8-week. It was controlled, double-blind, placebo-controlled. We did allow for 24 weeks of open-label NAC after the placebo-controlled portion of the trial. We ended up having 61 subjects complete the trial, 30 and 31 in each arm. And the dosage of NAC was very high. We started out with a dose of 8 grams per day, and we had a specific regimen for how we allowed decreasing of that dose based on side effects or complaints by the patients. In the end, the trial, during the 8-week portion of the trial, most of our subjects took the very close to the 8-gram dose because the median dosage was almost 7 grams per day. And the outcome measures were, as I mentioned, the glutathione levels measured in whole blood by GSH or in T cells by a FACS measure, which I'll talk about very shortly. Interestingly, we found some very significant results in terms of survival that I'll allude to, but those results we need to discuss a little because survival wasn't a formal outcome measure. This is just to tell you the beginning part of the trial, which before we even get to the results of the trial itself, we have some interesting results from the subjects who we screened for the trial. We did a telephone screen to start out with, and we screened over 800 subjects. Of those, I forgot to say that we're looking at people under 500 CD4 count. So that was part of the reason for the telephone screen, just to find out their CD4 count by phone. Then we took a laboratory screen, and this is laboratory tests that we did in our lab, including glutathione measurements. We had a fairly large number of HIV-infected people we could look at, well over 200. I want to tell you on the next slide some results we had from these subjects, first of all, before we get into the trial results itself. And let me mention here how we actually measure glutathione by FACS, which is one of the measurement types I talked about. We use the reagent monochlorobimane, which actually diffuses into cells freely and reacts with GSH inside cells to form glutathione S-bimane. Once monochlorobimane reacts with GSH, it becomes fluorescent. And also, once it forms this reaction, it stays inside cells instead of diffusing out freely. We do have to alter the medium a little bit to ensure that the bimane does stay within the cells. We add probenecid to the media, but for the most part it does stay within the cells, and it's a fairly robust measurement of GSH inside cells. And then this could be measured on the FACS. In addition, we can surface stain the cells to determine which subset we're looking at. So in this way we can determine what is the GSH level within CD4 cells, within CD8 cells, and so on. And here I'm showing what the comparison is between the measurement of GSH by FACS and the measurement of GSH in whole blood by HPLC. That's the left-hand plot here. On the y-axis is the whole blood measurement by HPLC, and on the x-axis is the measurement by FACS. And you can see that there is a fairly good correlation. Remember, these are quite distinct different cell types that we're looking at. Red blood cells, and which is the measurement in blood, and versus T lymphocytes.
Each dot is an individual.
Each dot is an individual, exactly. And the ones that are green are HIV uninfected, and the orange ones here are HIV infected. And the plots on the right are just showing all bimane measurements. On the x-axis is CD4 T cell GSB, and on the y-axis this is CD8 T cell GSB, NK GSB, B cell GSB, and monocytes GSB. And you can see that there's always a fairly good correlation, but when the cell types are more closely related, for instance, CD4 and CD8 T cells, the correlation is closer than it is with monocytes.
These are HIV positive or negative?
The ones on the right-hand side here, if I remember correctly, are mainly HIV-infected subjects. I don't think there are uninfected in there. But we have seen the same results if we look at uninfected. Yeah, we have. It's, you know. And then here in this slide, I'm showing you, just confirming that in HIV-infected subjects, the glutathione levels are lower. Either if you look at the measurement by FACS, by GSB, where the uninfected subjects, the distribution of levels is higher compared to the infected subjects, or by the HPLC measurement. Keep in mind with glutathione that there's quite a bit of individual variability. If you look at the distribution for the uninfected subjects, you will see some subjects who have very high GSB levels and some that have very low. And we continue to see that. There is just individual variability from person to person. So it makes it a little harder when you're trying to compare overall groups because you can see there's quite a bit of overlap.
There's some somewhat lay people here. They understand what the graph is, the bar graph of individuals.
It's a distribution, so it's showing the number of subjects on the y-axis here versus, you know, so many subjects fall within this range of GSB distribution. And let me point out here those subjects who were enrolled into our trial. I mentioned that we screened for trial entry to choose those subjects who were deficient in GSH to start out with. And the trial subjects are here in green. So we specifically chose them so they were definitely on the very low end of the control subjects. I'm hoping that in this case we'd be more likely to see an increase in GSH after treatment.
So why is there a difference in total GSH?
Between the uninfected and infected?
Right.
Well, that's been seen before as well. We'd expect that to be the case.
But that's principally red blood cells.
That's principally red blood cells, exactly.
Okay, and there's no difference in hematocrit, or there is a difference in hematocrit?
There's no difference in hematocrit.
So this is a red cell.
This is a red cell GSH, is lower in HIV-infected subjects. Yes. And this was a very fascinating finding that we found from the 240 individuals that we had screened for trial entry. We found that there was a difference in survival based on the level of the GSH in the CD4 T cells. Either if you look at the entire group of somewhat over 200 subjects, or if you only look at the group under 200. We feel it's actually better to focus on the group under 200 because that's the group that's more likely to die. I should mention the way we collected this data is that after the trial we continued to monitor the patients to find out if they were surviving or not. That's about the only endpoint we were able to determine. We weren't closely monitoring these patients, but we were following death records and other means to determine whether or not they survived. We monitored this for a period of 2 to 3 years after they had been screened with this very first measurement. But it is an unbiased sample because it's just those basically over 200 people that we followed. We weren't able to get survival data on all of those subjects, but for the group under 200, it's the vast majority we were able to get that data on. And you can see here that the difference is quite significant. Those who have the higher level of CD4 GSH levels survived quite a bit better than those who had the lower levels. I want to point out right here that this is only looking at the difference between CD4 GSH levels. And I don't have a slide to show it, but when you do a multivariate analysis, when you include other parameters, most notably the CD4 count, not only is this difference based on CD4 GSH levels still very valid, it's actually more significant than the CD4 T cell count. Now, other surrogate markers, such as the viral load, also don't seem to be as significant as the GSH, but I have to say, for that purpose, we didn't have a complete set of data for the viral load. So I can't say definitively that this difference will hold up as significantly here when you include viral load as a cofactor. Although I have to say we did have a fairly large number of patients who did have viral load data, and for that subset, the difference here with terms of the GSH in a multivariate analysis was still very significant.
Are the viral loads lower in the group with yellow?
And are the viral loads in terms of the correlation with the
More slow progressors in that group than there would be in the other one?
Yeah, I'm trying to remember now, and I'm sorry, I can't answer definitively if I can't remember the data exactly. There was not a very close correlation between the GSH levels and the viral load, if I can remember. I think there was some correlation, But let me hold off on answering definitively since I can't remember the data.
What about that whole blood glutathione, the red cell glutathione? Was there any correlation there?
The whole blood glutathione was only done on the trial subjects. So it was only done, and we only had 30 subjects in each arm, so it's a very small group of subjects. And even fewer than the 30 subjects we were able to do the whole blood on, for reasons I'll get to when I show that data. So we were a very, very small sample size. So we could not really analyze survival on those subjects based on whole blood. Just weren't enough subjects there to...
Since you broke down the patients into two groups based on just one measurement, in fact, on one measurement for each patient, do you have any idea about longitudinal evolution of GSH levels in the course of the disease among one patient?
Actually, unfortunately, we don't. We have not really done a longitudinal study here to follow patients over time. Did you want to?
But you could argue that if there is a correlation, a negative correlation, as the CD4 counts go down, the glutathione levels go down also. So it's not a longitudinal study, it's cross-sectional. But looking at the CD4 counts, the further the progression of the disease, the more likely you are to have low glutathione levels.
And I should point out, just to be complete, the break point that we chose here in terms of what number to use was actually determined by a statistical method referred to as the ROC analysis, receiver operating characteristic curve analysis. So it is an unbiased way of actually determining where best to determine to break this data. To return to this flowchart, to give you the results of the trial itself, after the screening process, we actually enrolled 83 subjects, randomized 41 to the NAC arm, 42 to the placebo arm. 2 actually had to be removed from the placebo arm. They were misclassified and just were removed from the analysis completely, leaving us with 41 and 40. And then we had 10 who did not complete from each side of the arm. So that was very well balanced. And then 31 and 30 from the NAC and placebo side. Baseline demographic and clinical variables just show that the randomization worked well. There were no significant differences between the two groups. I want to point out on this slide, though, one thing, antiviral drugs. At the time that this trial was conducted, the protease inhibitors were not introduced as yet. So the only drugs that these patients would have been taking would be the nucleoside reverse transcriptase inhibitors, AZT, and those types of drugs. And about 50% in each arm were taking some type of those drugs. And this slide very graphically shows that the main endpoint of the trial was successful, that GSH levels were elevated after 8 weeks of treatment with NAC. If you look here at the baseline values of whole blood GSH, this is the HPLC measurement, there's no difference between the placebo and the NAC group. But if you look after 8 weeks, you can see that there's elevation in the NAC subjects compared to the placebo subjects. For comparison, we're showing you here a group of HIV uninfected male subjects and what their GSH level is in blood. So you can see that NAC treatment for 8 weeks increases levels. It doesn't quite increase it up to the levels in uninfected subjects, but it's close. And the next slide shows you a more appropriate statistical analysis of this data, where we've done a model fit multivariate analysis, just showing you that it is very significant that when you have trial arm and baseline GSH in the model, predicting the change in GSH over 8 weeks, that you get a very significant effect for the trial arm. Both of these variables are very significant in the model. Now this here, I should point out that one of the drawbacks we had of our trial design, we realized that afterwards, is in order to do an intent to treat analysis, you need to include all your trial subjects in the analysis. We did not have an outgoing whole blood analysis for those subjects who withdrew from the trial. So in order to make up for that deficiency, we did a very thorough statistical analysis where we estimated the missing values. We actually have a letter into Lancet that explains our procedure for how we did this. We actually did a repeated series of estimations and repeatedly ran this analysis 50 times based on those 50 sets of estimated values. And we were still able to get a significant result, even with all those estimated values added back. So this convinced us that this, even with an intent to treat type analysis, is a very significant result. Missing values, yes. That slide was showing you the effect on whole blood GSH. We also see an effect on CD4 T-cell GSH measured by FACS. The effect here is much less robust, but it is still significant. The level of significance is P04, and in this multivariate model, we have baseline CD4 T-cell count and baseline beta-2 microglobulin as covariates in the model. Our trial also proved to us that NAC is safe in HIV-infected subjects. There's absolutely no difference in reports of symptoms or adverse effects between the trial arm and the placebo arm. The way I'm showing the data here is the median number of symptom reports per subject per visit, and just over 1 report per subject per visit in each side. I'm summarizing the types of reports are things like gastrointestinal upsets, diarrhea, nausea, things like rash, headache, fever, fatigue, stomatitis, depression, a lot of very variable kinds of reports. All of this data was collected because we're required to, but it's quite obvious to us that there's not an increased report of one kind or the other in one side. In terms of GI complaints in particular, we actually did analyze those separately since NAC is reported to have more GI effects, and there was no difference between the two groups, between the NAC or placebo arms in terms of...
What was your placebo?
Our placebo, these were fizzy tablets that you dissolve in water, and it was exactly the same formulation, just without the NAC in it. And they were essentially indistinguishable. So.
That was without lactose. Whatever vehicle the NAC was in.
Well, it had lactose.
Is it?
They had lactose in it. David discovered that.
Oh, did he?
Those tabs have, so we could have had some lactose intolerance. That may be one part of the reason for the problems.
But it was
the new ones, the new Pharmanac do not have lactose in them.
but the lactose was in both the drug and the placebo.
That's right. Yeah. And in terms of the dose reduction, in terms of the percent of subjects who requested dose reduction, was the same between the trial arms.
But that's 6.9 grams, not 6.9 milligrams, right?
Yes, that's a mistake. Thank you. Yeah, that's 6.9 grams. Yeah, I bet that's in our published paper, because this came directly from the published paper, but oh well. And I included this slide because last time I presented this, I forgot to point this out. Other surrogate markers, such as CD4 T-cell count and viral load, over the 8-week period of this trial, we saw no difference in terms of the effect of trial arm on these two markers. Of course, for CD4 T-cell count, this is a very short time period to expect to see a difference. In terms of viral load, there's no effect of trial arm. We did actually see a significant decrease in viral load in both groups compared to their baseline measurement. We attribute this as possibly like a placebo effect, or possibly due to the fact that this is a controlled conditions, and we had requested subjects to refrain from taking drugs or activities that could deplete GSH. For instance, alcohol and taking excessive amounts of acetaminophen. Those were prohibited during the trial, so, or at least excessive amounts were prohibited. So perhaps that's the reason the viral load was decreased. This is the survival results that I refer to that are not placebo-controlled, to start out with. again, I mentioned that we've been monitoring survival of these subjects, or we had been, for a period of up to 3 years after the trial had initiated. And it turns out that those subjects who had taken NAC, either during the placebo-controlled portion or during the open-label portion, survived longer than those subjects who did not take NAC. And it's a quite significant finding. The problem, of course, with this analysis is that we don't know how much bias could be within here because the two groups are not necessarily unbiased. Some subjects, in fact, most subjects continue to take NAC after 8 weeks open label. They had the option of switching over the open label up to a maximum of 6 months of open label, which would be a maximum of 8 months total for those in the NAC arm. Some others didn't.
It's a mixture of both.
Yeah, it's a mix. The people, exactly. So the NAC arm consists of those people. Yes. And
Those who didn't take it are called no NAC.
Steve, you gave us that roughly 80% of subjects went from each placebo control.
In terms of the open label, you want to go back to that one? Let's just go back to. Yeah, in terms of the frequency of the people who went on to open label, from the NAC arm, 90% decided to actually take open label. From the placebo arm, about 73% decided to take open label. The reason we included this on this slide in terms of safety, figuring that, well, perhaps, and side effects, perhaps NAC people, if they were having more troubles with their drug, would have been less likely to elect open label. But there is not a significant difference here, and if anything, more people on the NAC side decided to take open label. Who knows what that means? Maybe they were feeling better, and maybe they were more likely to want to take NAC for that reason. We like to think that, but obviously we can't say that. So the people who are not taking NAC in that survival analysis are those people from the placebo arm who decided not to go on to open label. That's the no-NAC group there, the majority of the no-NAC group.
Steve, did some people on open label NAC after the 6 months then continue to buy NAC on their own? You know?
We actually had asked our clinical manager of the trial to look into that. For most part, no. For the most part, they did not. I think we only had 1 or 2 subjects that we knew of that went out and bought it on their own to continue.
Yeah, you have to understand the psychology at that time. There were so many trials that people could enter into that the drug of the month or 2 months is the one that people would go for. I mean, they would switch from one thing to another.
Could you help me understand your survival curves a little better? Like after a while, the difference between the NAC and the no-NAC groups, and the survival diminishes, and is that related in any way? You said that they could take the NAC for only 6 months or whatever, or were they taking it for the entire period?
Yeah, it's hard to say. I mean, we think of it as this survival curve being shifted over. And you can almost think of it as being shifted over for the period of time that the people in general are taking NAC. That is one way to view the survival curve. It does seem to. In terms of, I apologize for the way of displaying the data this way is really an aberrant way. I should have updated the slide, and we've learned since from our statisticians that the curves crossing like this are meaningless. So you can ignore that part of the curve. But you're right. This difference in here happens to be about the time period that people were taking NAC. And again, it's interesting, but you know, I take this data very, very, very lightly. But it's interesting. It's
Steve, you do have CD4 counts. Probably will know. How does it correlate?
I'm sorry. What were you asking?
You probably have CD4 counts on these individuals.
Yes. I'm not showing the slide, but we as well did a multivariate proportional hazards analysis on this data, which is, again, the appropriate way of doing it. And again, you can include CD4 as a covariate. You can include viral load as a covariate. And in that analysis, NAC treatment, yes or no, is still the most significant parameter. So this is a nice visual way of seeing the data, but a proportional hazards model that we have done also shows that NAC is very significant.
If you plot that data as percent surviving as a function of the glutathione in the CD4, do the two converge? Do the two curves converge?
You mean a separate survival?
Only made at 0 time at baseline.
Because you don't have any other measures.
No, not of glutathione.
We have 8-week measure. We don't have.
Yes, we don't have, that's right, we don't have measurements after that. This is one slide I have of our newer data. We actually have been starting to collect some data from the Positive Care Clinic here, where we can now look at HIV-infected subjects who are being treated with the new HART therapies. That's the highly active antiretroviral treatments that include the protease inhibitors and the non-nucleoside reverse transcriptase inhibitors. Virtually all of the patients here in the clinic that we've looked at are treated with these new therapies. And the GSH levels, as measured with our FACS assay, are still lower in the HIV-infected subjects. So it appears as though this treatment is not completely replenishing the glutathione levels here. So glutathione depletion remains an issue.
Subjects who are not taking that medication?
Yes, we've determined that these subjects are not taking NAC.
And they've been on HART for months.
At least months, yes. Fairly long-term therapy.
Do you ask them to take them out
No, we actually don't. Sorry. Yes, we should. And we will. Yeah, that's exactly what I was just. I wanted to point out that this fits with recent data from Wolf Drogo's lab as well.
Actually, joking aside, there are, it's quite common knowledge, I think, in the chat rooms of amongst HIV-infected people that using SAMe could protect them from peripheral neuropathy and so forth. And so you may have some people in there.
No, I agree with you. I mean, we were thinking NAC is sort of out of fashion now among HIV-infected subjects, but I think SAMe is more in fashion. So it's, and we did not ask, and it's true. I just include this slide. There were 2 recent studies from Wolf Drogo's group over the last year that are very consistent with our study. One was showing that there is a massive loss of sulfur in HIV infection that's not improved with the new HART therapies. And there was another report where he reported 2 placebo-controlled trials looking at NAC treatment improving NK and T cell functions. And these were both quite significant results. In his placebo-controlled trials, he also did not find a significant effect on viral load. So NAC is behaving in a way that it's not an antiviral agent, but it is having a positive effect on the immune system. I'm just summarizing here our idea that, again, NAC treatment seems to be beneficial even in the era of HART treatment. I want to finish with some recent analysis that we've gone back to do on our trial cohort of people in terms of a relationship with treatment with AZT and glutathione levels. The treatments that our patients were taking, again, this is the placebo-controlled trial that I've been talking about, I grouped them whether or not they were taking AZT or not, because I found that the group of subjects taking AZT, alone or in combination with another NRTI, had lower GSH levels. This is a very interesting finding. So I'm just showing you here the types of therapies they were on. This is the group that I'm going to be comparing to the no treatment group. Remember, I showed before that about 50% of the subjects had no treatment at all in terms of antiviral treatment. And interestingly, the GSH level in whole blood was significantly lower in the subjects being treated with AZT compared to the subjects not being treated. The subjects treated with those other NRTIs, the GSH levels were not significantly decreased. Yeah.
The guess there is what? I mean, that those who are getting AZT might be worse or better.
Well, I don't know. It seemed to it may be specific to AZT.
Unless they were more sick.
Yeah.
That's what this slide is indicating. That it's not that case. At least it doesn't seem to be that, because if you look at the other parameters, it doesn't seem to be that those subjects who were taking AZT were sicker. Because even though the GSH levels are quite a bit lower. The other parameters that we measured are absolutely identical between the two groups. I mean, there's just no significant difference at all between these other surrogate markers. So it doesn't appear as though that's just simply the case. AZT is associated, of course, with the macrocytic anemia. That's well known, where although the overall hematocrit is the same whether or not you're treated with ACT, you have a much lower red cell count and a much larger mean cell volume. The reason I bring this up, it turns out that then if you look back and wonder, well, does NAC treatment affect any of these red cell indices? It actually does. It actually does seem to correct partially the red cell abnormalities that you see here. And the way we've done this analysis, it's the analysis is not extremely robust because we're talking about a fairly small group of people here. And we've divided up the group of HIV-infected subjects by their baseline level of GSH level. We've broken them down into thirds. And if you look at the subjects with the lowest GSH levels, you find that the group that took NAC had a decrease in their MCV, a move towards normality. And it's a significant result. So it's an interesting finding. Again, it's a very small group of subjects, and something like this would need to be repeated with a larger group of subjects. But it's just another positive benefit of NAC. And something to consider in terms of treatments overall, we should be looking at whether or not the newer treatments are affecting GSH levels specifically. We did a quick run-through in our current cohort, and again, I didn't bring a slide here because we don't have enough subjects to show whether something is truly significant or not, but we happen to notice, for instance, the group of subjects taking sequinavir has slightly lower GSH levels. It's actually significant, but only at the PeO4 level. So I think these types of studies are important to consider because if some drug therapies are specifically being associated with lower GSH levels, they should be accompanied by NAC to elevate those GSH levels back up towards normal.
Could you walk us through that?
Yes.
I don't really follow your main point.
Okay. My main point here is I'm showing a multivariate analysis here again, where we're looking at the change of MCV over 8 weeks. I'm sorry, is the mean cell volume of the erythrocyte. And remember, AZT causes an elevation in mean cell volume, and it causes a decrease in red cell count. And so the NAC treatment arm had actually a decrease in MCV, meaning that it's bringing it more towards normal, whereas the placebo group had an elevation in MCV, actually even increased over the 8-week portion.
Okay, so the AZT presumably was causing the increase in the placebo, and the NAC was preventing
and actually preventing that. Yes. And again, like I said, the numbers are small and the significant values are not extremely large, but it's intriguing considering that it is happening and we're seeing it even with those small numbers.
Is that placebo change realistic for being just an ongoing process of this ongoing increase in the mean cell volume untreated?
I don't know. That's it's a fairly, if you think about it, it's a fairly large change over 8 weeks.
What is that change in comparison to the difference between normal and the
yeah, I think you're
so
asking for this
90,
scale. Yeah, normal is about
over
90, and these are up at the
about 12,
yeah.
12, 13 units difference total, and they're changing by 2 over just an 8-week period.
Just over an 8-week period. So that's a fairly
Placebo,
large
that's-
change.
Sounds, yeah, a little questionable.
I should just mention all the people associated with this trial. Many of these people are no longer in the Herzenberg lab. Mario is now at the NIH at the Vaccine Research Center. I'm working with him. Michael Anderson is in Alabama. Greg Dubbs was our site director, and unfortunately he died secondary to a neoplasm related to AIDS. Stan Zarazinski is here. Hajime, you'll hear talking next. Steve Ela, of course, is here. Wayne Moore, David Parks. Malcolm Zaretsky is up at UC Berkeley, and he helped us with the statistical analysis for the trial. And then our current HIV study, we've been working mainly with Andrew Zalopa and Jose Montoya, and a new postdoc, and actually not very new, she seems new to me, but Moni Green, she's been here about a year or two. So thank you very much.
Are there any further questions people want to ask Steve?
If you took subjects that did not have glutathione depletion and gave them NAC, do you think their red blood cell glutathione levels would be increased on your methods of measurement? Or has that been looked at?
Hasn't been looked at, but I mean, our suspicion is that there's a homeostatic control of GSH levels. So if they don't have depletion to start out with, I wouldn't expect that they'd be elevated. So my feeling is that really, in terms of supplementation with something like NAC and HIV disease, it would be nice to do it in concert with a measurement. You measure the GSH level, and those that are known to be low, you supplement. But since NAC is safe and effective and really essentially harmless as far as we can tell, I mean, there's no reason not to recommend it, though.
What made you say that you think NAC has fallen out of favor?
Just when we've talking with some of the HIV clinicians, they tell us that their patients aren't taking it, as long as they know or not. I don't know how well they know that, but you don't believe that, okay. Okay, because that's my only really knowledge of it.
Here at Stanford, I mean, the positive care clinic, Andrew Zalopa says he knows what every patient is taking or not taking. He feels he has a very good rapport with them. And you know, whether that's true or not, I can't judge, but he... What?
Let's ask Diane. Diane, how many people do you know in Zalopa's practice who are taking?
I was just going to say, in Zalopa's practice, I happen to know a number of patients who are taking our trials, and I know that there are proponents of that. And he knows that well.
No,
I know he does.
He believes he's only got 2 patients.
Yes.
We're going to put this together, Diane,
Yeah, we are.
because this is really an interesting issue.
That is curious, because he told us that did not.
Okay.
Yeah, just one more question. What do you think is the mechanism? I mean, it's quite interesting to see that there's a relationship between NAC intake and the survival. And since there's seems to be no effect on viral load, do you think that NAC administration prolongs the survival of the individual T cells?
I think it's a very large question you're asking. And it's...
Maybe the next question...
Yeah.
Yeah, exactly.
Yeah.
So, NAC does more than just raise glutathione.
Yeah.
It deals with thyroidoxin, which is in fact the subject of the next talk. And so Hajime Nakamura has been back and forth between Kyoto and Stanford on several occasions, and on some occasions for rather long periods. He introduced into our, got us interested in thyroidoxin, which is another redox active molecule of very considerable importance, and it's one that the lab he comes from has been very interested in for, he can give an introduction on it, maybe he will, maybe he won't. But in HTLV1 infection as well as an HIV infection, as he's going to say, thyroidoxin levels in the plasma are increased. And Hajime gathered that data some time ago, and now this has been followed up on, and I'd be very interested to hear what some of the follow-up data is, because Hajime hasn't been here for about a year. Okay.
So, thank you. I'm happy to have an opportunity to give a talk here today. Next,
What's the picture of?
oh, this is so actually Junji's uncle is architecture, and they have the open museum.
Junji is his boss.
Yeah, yeah, my boss, Junji Adoi. Not Junji, but Junji's uncle. Okay, so let me start the brief summary of our cytotoxin discovery. So my boss Junji Odoi reported adult T-cell leukemia in 70s, and his boss Kiyoshi Takatsuki collected several cases in southwestern part of Japan, and they thought this is a kind of endemic disease. And after that, Gallo and Hinuma discovered the retrovirus HTLV1, human T-cell leukemia virus type 1, is a causative virus of this adult T-cell leukemia. And their colleagues, Takashi Uchiyama, made a monoclonal antibody against IL-2 receptor alpha chain in Waldman's lab, and they found ATL cells are overexpressing IL-2 receptor alpha chain. And in 85, so Junji Yodoi's group purifies the cytokine-like factor named ATL-derived factor, which can induce the IL-2 receptor alpha chain on ATL cells. But later, after the cloning of this molecule, ADF was human cytotoxin, an enzymatical, so they suppose it was a cytokine-like factor, but finally, it was a kind of enzyme. And actually, I joined his lab in 89 in this year, and for me, I was so curious. So why the enzyme-like factor shows cytokine-like function? That's why I continued to study this cytotoxin molecule. This is ATL cells, and ATL cells are very positive for cytotoxin ADF. Okay. And cytotoxin itself was discovered in Peter Reichert in Sweden, Karolinska Institute in '64. And as a cofactor for ribonucleotide reductase for DNA synthesis. So NADPH and cytotoxin reductase can reduce the cytotoxin, and cytotoxin transfers hydrogen for ribonucleotide reductase for DNA synthesis. And Arne Holmgren's group continued study this molecule. Actually, I moved from here to Arne's lab in '94. Okay. And now, cytotoxin system has another molecule, cytotoxin-dependent peroxidase. So even in mammalians, NADPH, cytotoxin reductase can convert this disulfide bond to reduce home cytotoxin, and reduced cytotoxin can reduce the protein disulfide bond to dithiol. Or in the presence of cytotoxin-dependent peroxidase, it is actually called peroxyredoxin family. And so far, we have already 6 isoforms in mammalian systems. One is in mitochondria specific form, and another one is secreted form, like that. And cytotoxin and cytotoxin-dependent peroxidase, peroxyredoxin can reduce the hydrogen peroxide. So cytotoxin system can act as an antioxidant system as well as glutathione peroxidase system in the cells. Okay, now cytotoxin has several biological functions. A variety of stress, including virus infection or inflammation, hypoxia, can induce cytotoxin gene, and cytotoxin act as a radical scavenger together with cytotoxin peroxidase, or cytotoxin can activate the transcriptional factors such as NF-kappa B and AP1. In case of the AP1, so cytotoxin can reduce the REF1 redox factor 1, and REF1 can activate the AP1 DNA binding. And NF-kappa B, cytotoxin can reduce the cysteine residue of P65 of NF-kappa B to activate, to bind the kappa B site. And
structure or the schematically of thyroidoxin?
oh, unfortunately, no. Oh, yeah.
Because it's...
Sorry.
Is there some...
This
You
is
can put
okay.
it there. I mean, maybe I should just summarize for you in
Oh, yeah.
quick word.
Anyway,
it's,
so-
it's a 12 kD molecule,
Oh, yeah.
a small protein molecule, which is normally found inside cells and has the activities which he mentioned.
So cytotoxin consists of 105 amino acids, and in 32 and 35 the cysteine residues. And between these two cysteines, they become so disulfide bond or diethiol form. So this is the oxidized form, and this is the reduced form. And it has another additional three structural cysteines. Anyway, yeah. So, is it okay?
So the business part of the molecule is these two cysteines
Yeah.
at 32
This
and
is active
35,
site.
which can form either a disulfide or a dithiol. So it's oxidoreductase. And now the story.
Okay. And interestingly, so since it was purified as ADF, it is secreted from the cells, and it shows cytokine-like function or chemokine-like function. So I would like to talk the first part. So the significance of cytotoxin in HIV infection. So it was done by my colleagues Mastani in Jundi's lab. So first we found in HIV infected people, the cytotoxin positive cells in lymph node were gone in AIDS-related case or it's a terminal stage of AIDS patient. And in vitro acute infection model with HIV virus, so cytotoxin expression is decreased after the HIV infection in acute infection model. So first, before I came here, I suppose cytotoxin level may be decreased in even in plasma of HIV infected people. But after that, we examined, so plasma level of cytotoxin were elevated in terminal stage of HIV infection. So this is the first study in Japan. And I was a postdoc here since 93 until 94, and I joined the NAC trial. And in the same time, I measured plasma level of cytotoxin in HIV infected people. And this is the negative controls. And in HIV infected people, those with rather higher level of CD4 cell counts showed a little higher level of cytotoxin. But in cases with lower level of CD4 cells, around one third of people showed higher elevated plasma level of cytotoxins. And in HIV-infected people, this here, so thyroxine level in plasma shows the tendency to be elevated in low CD4 cell counts. This is the negative controls. And thyroxine levels show the tendency to be invertly correlated with the monoclonal bimen staining glutathione levels in T cells. So if we divide it into two groups by 37, lower thyroxine levels, people with low normal thyroxine levels shows the around here, but people with elevated thyroxine levels shows the lower level of intracellular glutathione levels. So, sure.
Measure the form of the truncated form of thyroidoxine or both?
In this case, I use the ELISA system using two monoclonal antibodies, and this ELISA system can recognize only full length, not react with truncated forms. So, in HIV-infected people, as you know, so reactive oxygen intermediate induce the decreased level of glutathione, and in the same time, ROS, so systemic oxidative stress can induce the thyrodoxine, and most of thyrodoxine are oxidized and released into the plasma in the terminal stage of HIV-infected people. But, okay.
So do you know that it is the oxidized form that's released from the cell?
Not in the case of HIV infection, but after reperfusion injury, I measured most plasma levels of plasma thyrodoxine were oxidized home in Sweden. Yeah.
Both plasma thyrodoxine is oxidized. Is that the statement?
Yeah, we suppose, but we cannot so directly recognize the oxidized specific oxidized home from. We cannot distinguish oxidized home from reduced home thyrodoxine by antibodies.
But isn't there I mean, maybe we need Dean Jones or somebody else to ask this question. But I thought that all thyroidoxine in the plasma would be oxidized from the redox potential of plasma.
It should be oxidized.
Sorry?
It should be oxidized unless there's some mechanism to reduce it. But I guess that would, is thyroidoxine reductase, or is there something on the cell surface that could reduce it?
Beta's group already reported thyroxine reductase also secreted from the cells, and so even in the plasma, thyroxine reductase are existing. But no one knows NADPH can act in plasma. So if, thyrodoxine is oxidized in plasma, no one knows it could be reduced in plasma extracellularly. Maybe Beta knows something.
I just wanted to ask you about the dimer. What do you think about that?
Yeah.
That you can measure the dimeric form?
Yeah. So in Arne's lab, I measured most plasma thyrodoxine already became dimer when it is oxidized.
That's this,
That's you know very well. 37 one is the key player to become a dimer. Like this.
The crystal structure that Garth Powis was involved in, that's what they found the dimer, and they thought maybe that was the functional form, or involved in the function. But then later they decided that that's not.
No.
Yeah.
When it becomes dimer, it has no reducing activity. So this active site is buried inside of the dimer. Okay. But after that, so 2 or 3 years later, Lee called me, and yeah, Steve found, so this thyrodoxine was a very good marker for the survival of these people. And especially in case of people with low CD4 cells, less than 200. So, I mean, it's in immunocompromised host, elevate plasma thyrodoxine make it worse. So, individuals with elevated plasma thyrodoxine died more shortly than people with normal level of thyrodoxine. But we don't know why this plasma elevated thyrodoxine can get worse of this survival of these people in that time, at that time. And after that, as Lee's suggestion, I did the in vitro experiment. So question was, so secreted thyrodoxine can modulate the leukocyte recruitment in the inflammatory site. So thyrodoxine is released into the plasma, but might be this circulating thyrodoxine can affect the leukocyte extravasation into the inflammatory site. And I used this mouse air pouch model. So before the experiment, we inject the air subcutaneously on the back of the mice twice, 7 days and 3 days before the experiment. And on the experiment day, we inject the LPS, lipopolysaccharide, and 4 hours later, we collect the cells from the exudate in the air pouch. So this is a negative control using PBS and saline, and LPS induced a lot of neutrophils and lymphocytes. This is measured by flow cytometer. But just before the LPS injection, when we inject the thyrodoxine intravenously from the mouse tail vein, when I inject 40 microgram thyrodoxine, the blood concentration of thyrodoxine is like this. So during 4 hours, more than 100 nanogram per milliliter thyrodoxine levels were maintained. And the half-life of thyrodoxine is around 1 hour. But after 4 hours, when I collected the exudate from the air pouch, so neutrophils were gone. So thyrodoxine could inhibit the neutrophils extravasation from the capillary into the mouse air pouch. And it was the same in the case of thyrodoxine transgenic mice, where so human thyrodoxine gene was overexpressed by under the control of beta-actin promoter systemically. And in these mice, so in normally at resting time, circulating human thyrodoxine level were quite low. But after injection of LPS in their pouch, plasma level of thyrodoxine dramatically elevated. And during 4 hours, it was over 200 nanogram per milliliter. And this is a negative control in wild-type mice. So again, LPS accumulated LPS in their pouch, but in thyrodoxine transgenic mice, neutrophils were not collected.
Hajime, it would help to explain a few things. So the mice are transgenic for human thyrodoxine?
Oh, yes.
Okay. So they're carrying a human thyrodoxine, making it in the mouse. It's under a mouse reporter, or
no, human.
Human reporter, and so you definitely have increased levels of human thyrodoxine.
Yes.
And that's what you're measuring in that lower left box, right?
Yes.
The graph on the lower lower left. Yeah, that's human thyroidoxin.
Yes, yes.
Okay, and so the levels of human thyroidoxin are very low until you inject
LPS here.
LPS.
So LPS induces the secretion of human thyroidoxin into the blood. We cannot measure the plasma level of mouse endogenous thyrodoxin because this is the ELISA system using mouse monoclonal antibody.
So the human thyroidoxin is raised by giving LPS, and then you inject, at the same time you've injected the, what have you done about the air pouch?
Airport, so like before the experiment, so after 4 hours, we collected the exudate and measured the by-flow cytometer. So
let's look at the data now. So after 4 hours, you collected the exudate in the mice on the upper right. Yeah.
Yeah, this is the case of from thyrodoxin transient mice.
And so you have, although you have human thyroidoxin in the plasma.
Yes.
And that blocks the accumulation of neutrophils into the air pouch.
Okay.
So I assume mouse and human thyroidoxin are very similar. So that the human can take over
Yeah.
And the transgene is expressed everywhere, or in B cells, or?
No, everywhere. Yeah.
So LPS increases because certain cells get now activated and secreted.
Yeah, so we don't know the real source, but at least from the endothelial cells or other organ like liver, or they can secrete the thyrodoxin. Okay. And actually, so when we cultured activate neutrophils on the monolayer of endothelial cells, thyrodoxin, incubation with thyrodoxin can inhibit the adhesion of neutrophils on the endothelial cells. But this key system, two systems replaced with serine, mutant thyrodoxin, couldn't. So this inhibition was dependent on the active site of thyrodoxin. Okay. And also, I checked the initial marker of activation of neutrophils. And when neutrophils was activated by LPS, L-selectin CD62L was downregulated by LPS compared with the saline control. But when we inject thyrodoxin intravenously, after LPS injection, this downregulation of L-selectin was moderately inhibited. Or even in the case of thyrodoxin transgenic mice, after LPS injection, the downregulation of L-selectin was partly inhibited. So I think, and also, but when I measure the CD18 on neutrophils or VCAM1 expression on endothelial cells, thyrodoxin didn't change them at all. Even in the case of Hubeck or neutrophils were activated by LPS, even in both cases. So, yes?
Did you look at the lungs of your mice?
Pardon?
Did you look at the lungs of your mice?
Lungs.
Lungs?
Did you look at the lungs at all, or no?
How do you mean?
Because it's well known that when you inflict an injury to the skin, or even an intravenous, or maybe in the air pouch model, I don't know, there's one place where neutrophils are sequestered. It's in the lungs.
Yes.
And it's probably not CD62L dependent. It might be partially CD18 dependent, but not totally. So I was wondering whether there was an effect of the injection of human thyroidoxin or the presence of the human thyroidoxin gene in the mouse.
Later I can show you the dramatic effect of thyroidoxin on interstitial pneumonia. Okay. So from this data, I think this circulating thyroidoxin can inhibit the initial activation of leukocyte to start the rolling and adhesion. So by inhibition of shedding of L-selectin. But thyroidoxin cannot change the expression of L-selectin, LFA1 or ICAM1 expression, VCAM1 expression on endothelial cells or CD18 on neutrophils. But anyway, so thyroidoxin can inhibit the adhesion and transmigration.
Is that PDI?
Oh, yes. That's protein disulfide-isomerase.
So thyroidoxin doesn't affect the CD18 or CD11?
The expression of these molecules are not changed. So only CD62 L-selectin expression was changed by thyroidoxin.
And CD11B and CD18 don't affect the rolling of the adhesion of the neutrophils?
They do, but so thyroidoxin can inhibit the initial step, I think. So not this step. I mean.
So it's the pathway requires CD62L to be downregulated, and then
usually LFA1 are overexpressed on neutrophils and VCAM or ICAM1 on endothelial cells, and they play a role on the adhesion of these two cells. But thyroidoxin itself doesn't affect the expression of these.
We really don't know that, because of the PDI question that I guess Dean was alluding to also, that PDI can change it from a tetramer to a dimer, and that may or may not affect the staining intensity. So it could be that it's affecting it, but just not the staining intensity is affecting it.
Yeah, we should still study more details around here, I think. Okay. Anyway, so after this experiment, we suppose this oxidized thyroidoxin in plasma in HIV-infected people may inhibit the leukocyte transmigration into the inflammatory site, which cause more immunodeficiency in such an immunocompromised host in HIV infection. So that's why people with elevated thyroidoxin died more shortly, we suppose, in HIV-infected people.
People get that? They have criticisms of that? I mean, this is a hypothesis.
Yeah, this is hypothesis.
So I guess one of the questions is how is the oxidized thyroidoxin cleared from circulation? Because if it only has a biologic half-life of 1 hour, then there has to be a very...
They are secreted into the urine also, but we don't know the real metabolism of thyroidoxin. It is also secreted into bile from the liver. We should know the whole metabolism in the body, I think.
I guess what I was trying to think through was there's a fairly rapid pinocytotic exchange mechanism that you can account for a lot of things coming out of cells. But when you and also you would have dying cells that could be potentially releasing this. But when you start talking about the elevation levels that you're talking about, it implies that there is a very specific mechanism for it going out, and that it is not being cleared very efficiently.
Yeah, so secretion mechanism of cytotoxin is still unknown. So that's why, yeah, that's to be solved.
Or IL-1, or I think IL-6. I mean, there are a number of molecules which get out without having a signal or leader for secretion. But it's not due to dying cells. I mean, they get out of live cells.
Yeah.
That's an open area to try to understand.
Okay, but so in the case of HCV hepatitis C virus infection, so elevated cytotoxin doesn't affect the survival of these people. That's why I would like to show in this case. So this is asymptomatic carrier and chronic hepatitis, liver cirrhosis, and hepatic cellular carcinoma. So, so gradually increased cytotoxin levels in these HCV-related infected people compared with normal controls. And interestingly, so in the case after 2 weeks of interferon therapy, minus shows the viral load was negative, so HCV RNA was negative in the blood, and plus shows the still positive in the blood. So after treatment with interferon, so viral negative cases are most people with normal level of cytotoxin, but people with elevated cytotoxin levels, interferon therapy was not effective. And it is usually so interferon therapy, the indication was decided by the viral load in the plasma or viral type. But in the case of this data shows, so cytotoxin level is a quite good marker for the decides the indication of interferon therapy for these HCV infected people. And the ferritin level is also interestingly quite correlated with cytotoxin levels. So people with low cytotoxin and low ferritin, HCV infected people with low cytotoxin and low ferritin are quite good candidate for interferon therapy. It was, published last year in GSH. So I think even in the hepatitis virus cases, HCV hepatitis virus causes oxidative stress dependent on the iron ions. And this iron ion can induce the secretion of ferritin into the plasma, and as well, in the same time, cytotoxin is also released into the plasma. So if they are already high in plasma, interferon therapy is not effective. But if they are not so high, still so interferon can kill the virus in these cases, I think. And in these cases, so survival was not affected by cytotoxin levels. So anyway, so plasma level of cytotoxin can inhibit the transmigration of leukocyte. So we would try to use cytotoxin for more clinical therapy for inflammatory disorders. And before, so our colleagues already published after lung ischemic reperfusion injury using dog model, cytotoxin administration was quite effective for the preservation of lung, as well as NAC. NAC and ADF cytotoxin could keep the high O2 saturation in the lung compared with the normal 100 minutes warm after 100 minutes warm ischemia. And so recently we used the interstitial pneumonia or lung fibrosis model in mice. And this is the bleomycin-induced lung fibrosis model. So cytotoxin administration can block the lung fibrosis after bleomycin injection. This is in mice model. So bleomycin induced the infiltration of leukocyte into the interstitial space of the lung, but cytotoxin administration could inhibit this one.
Were you able to reverse those changes with, if you didn't treat simultaneously with thyroredoxin, did you reverse the bleomycin toxicity?
After, you mean?
After the injuries occurred, did you reverse it?
That's a good question, and we are now trying that. But so far, prevention was effective with cytotoxin, but not treatment after the development of lung fibrosis.
Always have to ask this question then.
Yes?
So would NAC not do the same thing as thyroidoxin
here? I didn't use NAC in this case, but yes, somehow, yeah, NAC also could be effective, I think. And even in the cytotoxin transgenic mice, bleomycin-induced lung fibrosis was dramatically inhibited in cytotoxin transgenic mice compared with the wild-type mice. And our colleagues, Dr. Hoshino in Kurme University developed this cytokine-induced pulmonary interstitial pneumonia model using IL-2 plus IL-18. So he injected IL-2 plus IL-18 every day, and this killed the mice within 3 weeks, usually. And as a negative control, we used ovalbumin as a protein carrier, but it was not effective. But when we inject cytotoxin every second day, could rescue the mice from the interstitial pneumonia. And this is the lung. So compared with this dead mice, cytotoxin injection could prevent the interstitial pneumonia dramatically. And even in the case of cytotoxin transgenic mice, around 80% were resistant to this IL-2 plus IL-18 induced interstitial pneumonia. So lung is a quite, so lung has the highest concentration, saturation of oxygen. So lung is the most sensitive organ for oxidative stress. And in this IL-2 plus IL-18 model, only lung was affected, but not other organs. So lung becomes interstitial pneumonia, but in other organs, they are apparently normal. So these mice are dead caused by this lung failure. But so cytotoxin could inhibit this infiltration of neutrophils into the lung interstitial space, and cytotoxin rescued the mice.
How much overexpression was there in this training? How much? I mean, was it just double the response or 10 times the response as far as normal, as far as what you would normally see the thyroidoxin responding to this treatment?
In cytotoxin transgenic mice, so enzymatic activity was roughly 5 times fold. Yeah. So I just finally clicked the recent our findings on cytotoxin transgenic mice. So this is the almost several times hold the enzymatic activity of human cytotoxin in this cytotoxin transgenic mice. So cytotoxic mice are quite resistant to a variety of oxidative stress. It is already published 2 years ago. So ischemia, brain focal ischemia, in wild-type mice, these white area are cerebral infarction, but in transgenic mice, the area was reduced by this model. And oxidized protein levels also decreased by protein carbonylation assay. And when we cultured the mouse embryo from day 7.5, we can culture the whole embryo in vitro system around for 2 days. And under 10% O2 oxygen concentration, they grew normally, wild-type mice. But under high concentration, like 25% oxygen, so many cases shows the developmental abnormality. But cytotoxin transgenic mice grew, most of cases quite grew normally under even this high concentration of oxygen. So systemically cytotoxin expression can prevent the oxidative stress-induced developmental abnormalities.
So you're going to tell us how? Are you going to hypothesize, make a hypothesis on how it's working?
How it works?
Yeah.
So at least as an antioxidant, cytotoxin overexpression can prevent the oxidative stress-induced.
In the plasma is oxidized. How can it be an antioxidant?
No. In this case, in the tissue levels, cytotoxin. So in the cells, cytotoxin are reduced home. Most of cytotoxin could be reduced home in the presence of NADPH cytotoxin reductase.
So thyroidoxin acts as a catalyst to bring reducing equivalents to this tissue, or perhaps in the bleomycin situation.
When it is secreted, so no one knows it is cytotoxin is oxidized home or reduced home. But in the cells, so intracellular cytotoxin should be reduced home.
And there, of course, is a continual reducing system,
yeah.
Thyroidoxin reductase.
Yes.
And
so in the presence of supplying enough NADPH.
So again, I guess in this situation, if, well, no. In this situation, I don't know. I mean, you wouldn't expect to give NAC or glutathione having
Here, maybe, incubation with NAC may be effect. We don't know. And sorry, Japanese, but this is just mentioned the name of our collaborators. So, cytotoxin transgenic mice, so bone marrow cells are quite resistant against UV light. So this is a colony formation. And so this is normal mice, and this is bone marrow cells from transgenic mice. So under the UV radiation, still so many colonies are formed. And also, so finally, cytotoxin transgenic mice can survive longer than control mice, around 25%. So they are quite resistant to several kinds of oxidative stress, and maybe less sensitive to several signals, but they can survive longer. Yeah, maybe I showed you before.
I know Junji has reported this several times. He was going to repeat that experiment
Yeah, but the repeat of this experiment takes 2 years. So, so not yet. We don't have the new data. Okay, and cytotoxic transgenic mice are more resistant to Listeria monocytogenesis infection. This is the survival data, and after 1 to 3 days, this is the colony formation unit of Listeria in spleen or liver. So in cytotoxic transgenic mice, so Listeria cannot grow in these mice. But in vitro study showed there is no difference of killing activity of macrophages from normal mice and cytotoxic transgenic mice. So we don't know the real mechanism of this inhibition of Listeria proliferation in these mice. But anyway, cytotoxic transgenic mice are quite resistant to infection model. So still we have several remaining questions. So we don't know the real mechanism of secretion of cytotoxin. And it has some extracellular function. So it should have some target molecule on the target cells. And we should know the signal transduction mechanism via exogenous cytotoxin. And from this April, we start a new center named Human Stress Signal Research Center. So we should develop here new signal transduction mechanism and stress monitoring system or stress attenuation therapy in this center. And this is the new building for our collaborating hospitals. Okay, so this is our collaborators. So Steve and Lee Ben are here. So thank you very much.
Steve didn't show this data, I thought Hajime would show it. But when we look back in our clinical trial at the patients who were treated with NAC compared with placebo, that we had thyroidoxin data for, and again, it was a subset of patients because we didn't have all the thyroidoxin data, but nonetheless, there was a significant decrease in the thyroidoxin levels over the 8-week trial period in the patients who were treated with NAC. It was frustrating, this really wasn't enough data, but it definitely was there.
You showed us all of these data in the transgenic mice where you had protection. It's equally interesting what you tried that you didn't have protection. Was there anything?
So in the case of HIV infection, so in immunocompromised host, so this high level of plasma cytotoxin get worse. But
in the transgenic mice, you challenged the transgenic mice with many different types of insults, and it protected the transgenic mice were protected against the listeria, against the hyperoxia. Did you try anything that was not protected?
Okay. For example, acute acute hepatic failure caused by L-galactosamine plus LPS was not protected in this mice. Maybe it's usually the galactosamine sensitized LPS injection caused acute liver failure within 6 hours. And this might be more acute, too acute model for prevention by cytotoxin. So we showed rather moderately slow subacute model in these cases.
A more chronic model might be urine AIDS.
Yeah, that's
Consider trying that?
I'm not yet, but
That model is a total mess.
That's a hard one to work, yeah.
Well, I don't care what you call it, Lee.
It's
But,
it's
well-
a dreadful model, Steve.
Well,
It's not
let's
what I
not
would
consider it a
recommend.
model. Do you think
yeah,
that,
even in our institute, some other scientists are studying maize. Might be.
The models that you've chosen here and that worked were all ones that have been shown previously to be protected by antioxidants. Correct? I mean,
Most cases.
and listeria, which moves the Th1 versus Th2.
I don't know that embryonic culture model, but
Yeah, I mean, it's got to be an oxidative stress model, and whether they've generated a protection model for it previously or not, I don't know. But if you gave NAC or glutathione, I mean, that would protect against hyperoxia. So each of these is just so that I guess thyroidoxin is acting. When you have a lot of thyroidoxin acting as a catalyst to bring reducing equivalence to the system that's being damaged, being put under stress.
yeah, of course, as an antioxidant system, glutathione is roughly some millimole order. Thyrodoxin level is micromole order. So 1,000 times glutathione is the major component as an antioxidant system. But for example, in the activation of transcriptional factors, thyrodoxin sometimes 1,000 times more effective to activate the NF-kappa B activation compared with glutathione.
Use a lower concentration of it. Yeah, because well, that's true.
So in some way, thyrodoxin.
Yeah, but you can't. I mean, so I guess you're not going to give. I mean, for people, for treatment of humans, you're not going to make them transgenic for thyroidoxin at the right time. I mean, they're going to be dead, long before that comes up. But you can treat them with NAC, for example, or with glutathione, and get the consequence you want. I mean, protection.
Yeah. How about the combination? NAC and thyrodoxin.
Well, I don't know how much you have to inject.
High plasma is worse.
Hajime said that it's working intracellularly.
How do you get it?
Yeah.
The problem essentially is that the situations in which you want to protect with thyroidoxin are acute situations, stroke, ischemic injuries. Delivering thyroidoxin, I think Hajime is right, probably a combination of NAC and thyroidoxin might be even better. Because thyroidoxin won't clean up oxidants that are there, whereas NAC or glutathione would.
We are also trying to find thyrodoxin inducer. And recently granule acetone can induce thyrodoxin. Geranil acetone.
Geranil.
Yeah, it's already clinically used as an anti-ulcer drug. Maybe my pronunciation was...
Yeah, Geranil, G-E-R-A-N-Y-L. A-N-Y-L, yeah.
It's an isoprene unit, isn't it? That is an intermediate
Yeah.
in the synthesis of cholesterol.
Yeah.
GSH.
It's nothing Myers works with. Yeah.
It's already reported to can induce the heat shock protein like HSP72, and we checked the effect of GGA on thyrodoxin induction, and it can induce thyrodoxin. Acetate. No, acetone. Yeah. I don't know the chemical compound. Oh.
Geranil?
Yeah.
So then it's a geranil unit on each side of an acetone.
Don't ask.
Anybody else have any comments, questions?
How about heat shock? Will heat shock induce thyroid oxygen?
Will heat shock induce thyroredoxin?
Heat shock. Oh, heat shock doesn't induce thyrodoxin. So thyrodoxin promoter contains several responsive elements, including oxidative stress responsive element and xenobiotics, XRE, xenobiotics responsive element, or ARE, antioxidant responsive element, and hemine responsive element. But the heat shock directly doesn't induce thyrodoxin.
As I understood it, the thyrodoxin is a phase 2...
phase 2 detoxifying enzyme, yeah, a kind of.
But also, no, but at the end of I'm sorry, I was trying to say the right thing. It's one of the late-acting secreted proteins when you have
infection.
Infection.
It's a.
I can't think of the word. I'm sorry. It's stupid. I've lost the word. But anyway, it comes out late when you're having septic shock, and my own supposition is that its purpose essentially is in a case where you have infection, that towards the end of that infection, you have to downregulate the influx of neutrophils and downregulate the inflammation, and that its normal function is to be released at that time. And if you don't release enough of it, then you go towards septic shock and you start building up. Does that make sense to you?
Yeah. I agree with you. Yeah. So in some sense, thyrodoxin is a endogenous responder to such an inflammatory process, I think, to attenuate the more inflammation. Yes.
Did you make a thyrodoxin knockout? I thought I saw
Yeah, thyrodoxin knockout mice are embryonic lethal.
Yeah. Thyrodoxin you need in order to synthesize deoxynucleic acid.
Yeah, okay.
It's a very central molecule.
But is that true? I thought there were multiple forms of thyrodoxin. Is that true that that just knocking out one form, or do you have to knock out multiple forms?
We have only one gene, but so in protein level, it becomes truncated forms, and like here, around 80 or 84. Total is 104 or 5. So this truncated form has no reducing activity, and it shows more cytokine-like activity, including mitogenicity. Or, Vita is expert.
It doesn't work to block neutrophil migration, does it, the truncated form?
Truncated form are more chemoattractant for eosinophils. It was already reported, but for inhibition assay, I've never tried, because I don't have the truncated. Maybe, I can.
What I was referring to was that in the gene databases there are three. I think they list 3 thyrodoxins.
Human. In mitochondria, we have mitochondria-specific thyrodoxin. It names thyrodoxin 2, and it has a mitochondria translocation signal peptide ahead of this thyrodoxin, and it's around 60, and it is cleaved.
Yeast that has 3.
Oh, yeast.
Yeah. But am I right in understanding that there is no therapeutic application there is no therapeutic application, is that right, in giving thyrodoxin? It doesn't get into the cells, you can't administer it.
Well, he's asking whether there is thyrodoxin therapeutically, do you think of it as something to inject?
For therapy. Yeah, so if we can make, recombinant thyrodoxin, we can use that.
Can I answer that question? He's asking you, will you use it for therapeutics? And they are working on using it for therapeutics in acute redox situations like stroke and heart attack, places where in fact you have a very rapid infusion of neutrophil, and it's quite effective in these situations. They have a dog model for this, and Hutchinson can tell you all the models. That was the question he was asking.
Yeah, we are trying to use. Okay.
It's all exogenous, and it's probably based on inhibition of neutrophil and inhibition of oxidative injury at the site, rather than intracellular.
Okay.
Is it really true that thyroxine cannot go into the cell?
So far, we don't have any direct evidence thyroxine can enter into the cells. So we don't know yet.
I don't know if I should, but staining murine cells with human thyroxine then stain for the human thyroxine. I found that it actually goes into the cell.
Yeah, it goes into the cells? Oh, really?
I couldn't see any staining in the mouse cells when incubated with thyroxine, and I could stain it with active human thyroxine.
So in your case, how you can detect that it was intracellular by staining?
Looked in the microscope. It was intracellular staining with antibodies. But
so after you made cells permeabilized, so you fix the cells?
Yeah. Yes, of course.
How do you know it wasn't just stuck to the surface?
Oh, it was quite spots in the cells, actually. But, I mean, that was...
I don't know how you can see that, because it's a very hard problem always when you have the surface molecules and you look by the microscope. You can't really tell whether it's inside or out. It's a hard...
yeah, at least on cell membrane, cell surface, the thyroxine can bind some target on the cell membrane. So how you could distinguish that it was intracellular or on the cell surface? That's maybe we should radio label.
Yes.
Well, you do it without the permeabilization, and see if you can still see just the exposure, but no permeabilization gives marking, and it's probably on the surface. If you require the permeabilization, it doesn't happen without it. Then it's got to be inside.
That was actually what I saw, but it was one cell.
It's all right. I mean, if you show that you can't see it, that you have to permeabilize to see it, that is a reasonable argument that it's inside.
Yeah, that was a couple, I mean, I did it once, honestly, but that was what I saw, nothing in it.
I have one more question about the thyrodoxine knockout. Do you actually get ES cells then? If it's that important, then? So the ES cell, you get homozygous knockout ES cells?
And around day 6.5. So after implantation,
they can synthesize the DNA without needing.
Like Replenishment knockout. Okay.
Which day did you say?
I can. Replenishment. So
Well, I guess this is the end of the symposium, not the end of the festivities, I suppose. And we'll see you later or whenever. Anyway, I want to thank you all for coming and thank the speakers very much for their contributions.